So here I'm looking at the eye, and not just the human eye, but vision in general. So on that topic, what is vision? Well, vision begins with the capture of light energy by photoreceptors. Many invertebrates have simple visual systems in the sense that their photoreceptors are clustered in an eye spot. They perceive light's direction, but not necessarily a visual image like us. And you can see here, a nice, very simple, very basic to very complex development of the eye. Now there's 4 phyla that have evolved well-developed image-forming eyes: annelids, mollusks, arthropods, and vertebrates. This is our flatworm that has just the eye spots, and while they may look like very distinctive eyes, they're simply only able to tell light from dark. Eyes are strikingly similar in structure, but are believed to have different evolutionary paths amongst all of these phyla. Eyes of animals, as we see here, we have our insect eye, very basic, very large area of reception. We have the human eye of the vertebrate compared to the octopus. In initial inspection, if you look at both of these, they look very similar. However, what's distinctly different is the number 4 region. The 4 is the blind spot. In our eyes, we have an area within each eye, very small, but it's a blind spot. And what happens here is there's lack of photoreceptors. However, in the octopus eye, there is no blind spot. The retina continues the entire way. And 3 is the optic nerve, and that's what's sending all of this information of the image to the brain for processing. Continuing on, the structure of the vertebrate eye. Eye works like a lens-focused camera. We have the cornea located here, and that's a transparent covering that focuses light. We then have the lens, which completes the focusing, and ciliary muscles that change the shape of the lens. They're located here, and they can help pull or kind of condense down that lens so that the image can be properly focused on the back of the eye where the retina is located. The iris is a shutter that controls the amount of light allowed in. See that evident here. The pupil is a transparent zone located directly in the center. The retina is at the back surface of the eye, and the retina contains 2 types of photoreceptors, both rods and cones. The center of the retina, the fovea, is what produces the sharpest image. The fovea is the area that's producing that sharpest image. We want that focused on the back of the eye, because that's what's going to allow the image to be perceived in its clearest form. Now, rods are very sensitive to dim light, as we see here. They cannot distinguish colors, nor can they detect edges of images. They produce a poorly defined image. In contrast to that, cones can detect color, and they're able to detect edges well, and they help produce very sharp images. Rods and cones, both of these are located at the rear of the retina, not the front, because this is where our image will be focused. If it was in the front, that's where our lens is. That image then gets projected in the back. Now, color vision, there's 3 kinds of cones that exist. Each absorb different wavelengths of light. We have our blue, our green, and our red. So despite only having 3 cones, we're able to perceive many different colors. So these many different colors we're able to perceive is a combination or a blending of what these cones are receiving. We see that evident here with our red and our green. When they mix together, they form yellow. Now, if we mix red, green, and blue, we get that white light evident here. And a combination of these, it shows kind of the distinctive colors here, are how we're able to get these general wavelengths of light, and this is how we're able to perceive all of these variations of colors. Now, if you didn't see any colors there, you might be colorblind, which is a condition where a person cannot see all 3 colors. It's caused by a lack of 1 or more types of cones. It's inherited as a sex-linked trait. It is more likely to affect males because it's an X-linked recessive inherited trait. 1 in 12 men will be infected on average, and 1 in 200 women will be afflicted by colorblindness. If you see here, if you see just a bunch of dots, you might be colorblind. You should see a 74 here. So there's a 74 was the number for this colorblind test. Now, continuing on with the eye, we're focusing for distant vision. We're looking in the distance for things. Light from a distance needs very little adjusting for proper focusing. Far point vision, the distance beyond which the lens does not need to change shape to focus, is about 20 feet. Closer than 20 feet, we call that close vision. In the closer source, as the light is bent more sharply, by the relaxed lens to strike the retina. So we see both of these striking the distinctive back of the retina. Here for distant vision, our lens is very flat. For close vision, our muscles have adjusted to cause that lens to be fatter, bending that light so that it directly hits the back of our retina for a clear image. Focusing for close vision, we have accommodation, which is the changing the lens shape by ciliary muscles to decrease refractory power. We see that here. See that fattening of the lens causing, again, that focus point to be directly on the retina. Constriction is the pupillary reflex constricts the pupils to prevent divergent light rays from entering the eye. If you've ever had your eyes dilated after going to the eye doctors, this is what it'll look like. Pupils will dilate, and this is an eye here that did not have pupil dilation. Convergence is the medial rotation of the eyeballs towards the object being viewed. This is kind of a distinctive case here where this person is able to independently move one eye compared to the other. But to focus on a close object, both eyeballs are going to converge to that midpoint there. Problems with refraction. So if you wear glasses, this is important to you. If you have normal eye vision, that's with the light focused properly. Nearsighted, myopic, is the focal point is in front of the retina. So we see here is evident of nearsightedness. That focal point is in front of the retina. That means that when the image does hit the retina, it's blurry. How does that look? Well, you see here's a clear image, and here's kind of the near objects being a little bit better in focus, and those just slightly further away here, not having quite as distinctive kind of sharpness to that image. Farsightedness, the focal point is beyond the retina and corrective lenses with a different type of lens as we see here. So when we say focal points behind the retina, we see that here, indicating the image here is still blurry. These are corrected farsightedness with a convex lens, you see here, and nearsightedness with a concave lens. And that's adjusting so that in both cases now the focal point is directly on the retina. Types of lenses found in glasses, there's a bunch of different types, more than just for near and farsightedness. Depending on your vision, you may have multiple lenses in your glasses to help with both near and far focus. Bifocals, as the name implies, there's two different types of lenses, where you have one for far, typically located towards the top of the lens, and near for the bottom portion. Trifocals, as the name implies, there's three. There's a far, a middle, and a near. And there's also progressive lenses. They change magnifying power near to the middle to far vision gradually. They don't have these distinctive lines as a trifocal or a bifocal would have. In general, their purpose is the same, to help the individual see clearer. This is the image of Seattle being refracted through myopic glasses there. Binocular vision. Primates and most predators have eyes in front of the head. The two fields of vision overlap, allowing the perception of 3D images in depth. Prey animals generally have eyes located on the sides of their head. This prevents binocular vision but enlarges the perceptive field of view. We see that here with our pigeon. These pigeons have a very large field of view because they're often preyed on. Owls are predators, and they have not quite as large a field of view, more overlap, more of a blind spot, we'll say, because they're more focused. And these two fields that overlap, allowing perception of 3D images in depth. If you ever want to try this, you take both of your hands and put them out in front of you, and go through and touch the tips of your fingers. But then if you close just one eye and try to do it, you'll often miss because you lack that 3D depth perception. Lastly, some animals, chameleons in particular here, can move each eye independently, and this can allow for a greater field of view. They can focus both eyes and select prey before striking. So this is another kind of unique adaptation that some animals have for independent eye movement. Hopefully this was helpful in giving you that brief overview about the eye.